The nucleus contains the cell's genome and is the site for the synthesis of DNA and RNA molecules. The nucleus is separated from the rest of the cell and the cytoplasm by its nuclear envelope. This is a double membrane structure, perforated by nuclear pores, which in turn act as gateways, allowing macromolecules to move both into and out of the nucleus. The nuclear envelope has two distinct components, an inner nuclear membrane and an outer nuclear membrane. They exist as one continuous membrane, but nevertheless have distinct protein complements. The inner nuclear membrane is the binding site for the chromosomes and the so-called nuclear lamina. The outer nuclear membrane forms a continuous structure with the endoplasmic reticulum. The outer membrane of the nuclear envelope is physically continuous with the endoplasmic reticulum, or ER. The ER is a large network of interconnected flat membrane sacs, sometimes accounting for as much as half of the total amount of membrane in the eukaryotic cell. The layered structures of these membrane sacs are called cisternae, and the space within each cisternum is called the lumen. ER can be categorized as either rough ER or smooth ER. The rough endoplasmic reticulum appears as a rough surface under the electron microscope due to the presence of ribosomes on its membrane on the cytoplasmic side. These ribosomes on the ER are responsible for synthesizing membrane proteins, and those proteins that are destined for secretion from the cell. The proteins synthesized by these ER-bound ribosomes either accumulate in the membrane or in the lumen of the ER. These proteins are then transferred to the Golgi apparatus in order to be directed to their ultimate target locations in the cell. The smooth endoplasmic reticulum does not contain ribosomes and does not play a role in protein synthesis. Instead, it is involved in the synthesis of the lipids and steroids that need to be redistributed to other membranes inside the cell. The Golgi apparatus contains many flattened compartments that are stacked on top of each other. Structurally, the Golgi resembles the ER, however, the compartments are not interconnected. The Golgi is responsible for packaging proteins for secretion and synthesizing complex carbohydrates that are then expressed on the surface of the cell. The Golgi stacks accept vesicles that bud off from the endoplasmic reticulum. These vesicles contain secretory proteins that are further modified in the Golgi before being sent to their final destinations. These post-translational modifications include protein glycosylation, which is the attachment of a carbohydrate to a protein. At any given time, vesicles fuse and bud off from the Golgi stacks continuously, such that the Golgi is a very dynamic organelle. The Golgi apparatus holds a special place in my heart, in part because it is so dynamic. So in my mind, I've always seen it as this structure that's constantly changing, with vesicles being added to it, vesicles being released, and with that beautiful stacked structure that you can see in the microscope, I always wanted to imagine in my mind's eye what the Golgi apparatus would look like. I had the opportunity to do this, and what you're seeing right now is my visualization of what the Golgi looks like. And I think you would agree it is a thing of beauty. Lysosomes are small organelles that are less than 1 micrometer in size. Despite their small size, however, they have a big function. They contain digestive enzymes such as hydrolases. Proteins, carbohydrates, and fats are therefore broken down in the lysosome by these enzymes, and they provide the cell with simple nutrients and other critical components. However, if the lysosome did not sequester these enzymes from the rest of the cell, they could also degrade components of the cell, which would be fatal. Lysosomes therefore store these enzymes while also passing the nutrients through their membranes into the cytosol of the cell where they are needed. Mitochondria play a critical role in providing the cell with the chemical energy it needs to power its many reactions and processes. They do so by converting the energy found in food into ATP, the most commonly used molecule, that readily provides energy to perform work. One can think of ATP as the primary energy currency within the cell, and the mitochondria enable the conversion of various high-energy molecules, such as sugars or fats, into ATP. Since the primary function of mitochondria is the generation of this high-energy ATP molecule, mitochondria are often thought of as the powerhouses of the cell. Despite what we often see in diagrams, mitochondria aren't just static kidney beans that sit around in the cytoplasm. They actually move and are dragged by motor proteins on cytoskeletal filaments throughout the cell. By this mechanism, mitochondria can be localized specifically in particular regions where there is an urgent need for high levels of ATP. Each of the organelles we have discussed, the nucleus, the Golgi, the lysosome, and the mitochondria, can be thought of as a functional module within a eukaryotic cell. Each organelle, as we have seen, has a unique structure and chemical composition that is designed to perform a particular cellular function. We will begin a more in-depth investigation in the next few lessons of mitochondria. How does all of the components and the physical structure of a mitochondrion collaborate to actually result in a specific task being successfully executed? Namely, the task of synthesizing large quantities of ATP. It is important to keep in mind that even though the intracellular organization of eukaryotes may be more obvious, given all of their discrete intracellular compartments, prokaryotic cells also organize certain biological processes and chemical reactions to particular spatial regions within the cell. So how do all of these dynamic parts of the eukaryotic cell work together and in concert? Let's now take a closer look.